Okay, well, hello everyone, and it's wonderful to be back. Sonia Marshall-Gradisnik sends her apologies. We're very busy in the lab at the moment, and so she was unable to come, but she did ask that I convey her apologies. I'll make other acknowledgements at the end of the talk. So you will have heard some of this if you've been to the last couple of meetings, but we're building on the experience and knowledge that we've acquired previously. So you will understand that we're dealing with a group of non-specific calcium ion channels called transient receptor potential ion channels. And these are incredibly important for bringing calcium into the cell, and these are calcium ions. And calcium ions are absolutely critical in a whole range of functions, biochemical functions, mitochondrial functions, respiratory functions, and so on. And today we'll be presenting data on our findings of one of these channels, TRPM3 in natural killer cells, and also in the gastrointestinal tract. Now, there's a long history of research into natural killer cells in CFSME, and this goes back about 30 years. So, I have explained this previously, but to be sure that natural killer cells we use as a model in the laboratory because it gives us an enormous amount of information. So please don't go away thinking this is only a disease of natural killer cells. What you see happening in these cells is probably happening in every cell in the body that carries these ion channels, and we can talk more about that later. But it does lead to immune dysfunction. It leads to impaired function of the natural killer cells. And the function that we're talking about is what is their day job, and that is to destroy cells with bacteria and viruses and cancers. So it's very important when you do research of this nature, you can actually demonstrate what is the impact on function. If you just report certain findings in the cell, it's got a kind of a so what effect. So it's very important in the lab that you actually measure how well the cell is continuing to function or not function, and that's critical to this research. The other problem is accessing tissue. Obviously very difficult to access tissue in the brain, the heart, and so on, where all these receptors are located, but you can do that from the blood, and that's why we use natural killer cells. And it's very important that those cells are fresh when we do calcium studies. They cannot be frozen, the results would be meaningless. So what happens, and we have a wonderful group of patients who help us in providing blood samples, and it's really difficult for them because we need to take the blood at 8 o'clock in the morning. That's often a really tough call for patients. And then that blood is immediately taken to the lab. Everyone standing by in the lab goes straight into the lab, and they're working on it by after 12 o'clock. And then they'll work through the night, and then they get home about dawn the next day. So just to be really clear that what we're showing here, yes, it's in the immune system, yes, it's in natural killer cells, but it's probably happening in every other cell in the body that carries these sort of receptors. Now you can see the DNA, and up here we have things called SNPs, single nucleotide polymorphisms. So if you imagine this is a ladder that's just been rotated on itself, these rungs here are base pairs, and they have nucleotides as part of that component that joins the DNA sequence. And when there's a change or a change of expression in any of those segments of the DNA that transcribe genes or that reflect genes, then it's quite possible there's going to be a change of function. It's not actually as simple as that. You can have synonymous or non-synonymous SNPs, which means if they're synonymous, there's actually no change in the function, so the function of the transcribed area or the protein is still the same, so not a problem. But if they're non-synonymous, it means the function of that protein has been changed. And if that protein happens to be a receptor or an ion channel, then you're in serious trouble, because it means that receptor has changed and is no longer functioning, transmitting calcium as it should. Now, the other tricky bit about this is TRPM3 has more isoforms than any other TRP, and we'll talk about that in a minute. But I want you to bear in mind that if there's up to 12 different subtypes of the TRPM3, then we could be talking about a mix of any of these. And what's really important about that is one of the subtypes, called the TRPM3 alpha 7, is actually a non-functional. It's a dud receptor or a decoy receptor, and that would be bad enough, but it also contaminates the function of other TRP channels nearby. So you could envisage, and this is speculation, but if that one was ramped up, it could cause detriment to other TRPM3 subtypes, and therefore overall become non-functional. So that's just one way of thinking this through. But anyway, in here we've got the transcription into the protein of the ion channel, and as you can see, the calcium is passed through the ion channel, and that's what it's supposed to do. Now, in natural killer cells, there's up to 5 different phenotypes of natural killer cells based on the CD56. So this one's a CD56 bright, and this one's CD56 dim, and generally they mature from this one to that one. And you can see there's a number of functions in there. You've got a number of signalling pathways, that has important implications for the cell. And this one here is very important because it excretes toxic molecules which perform that assassin function, which, as I said, is a stage of. And we measure that by how much or how little it does that assassin job, and that's called lysis. So we put them in with a cell line, like a cancer cell line, and we can actually measure the extent to which it causes lysis or cell death of the cells in which it's exposed. And you may be familiar with this, but what we did was to extract the DNA of the natural killer cells and look at the genes that have the mutations, which are here, the SNPs, and this is their occurrence in both patients and in controls. And then you see what the odds ratios are here. So in other words, if you've got this particular mutation, then that's the likelihood that you will develop the illness, because this is taken out of ME/CFS patients and healthy controls. Now, in the top 15 or 16 of these odds ratios that we see here, you can see that as many as 5 of the TRP are TRPM3. So that was pretty important to us to say, well, something's clearly going on with multiple SNPs, hence the term, polymorphisms, occurring in the patient group. And this is the family of TRPs, transient receptor potential ion channels. There's 7 families, 6 in humans, and they're also called threat receptors because their job is to convert threats in the external environment into signalling activity which helps the body to adapt and cope and deal with those sort of threats to which it may be exposed. And this is the family, the blue family here, the TRPM3, but you can see there's a number of others as well. And this is how they work. So here are the threats here. And if you go down the list, you might see that many of these threats are what people with ME/CFS report as a problem. So when you see osmotic pressure or stretch or vibration, anything to do with physical movement, exercise, travelling in cars, planes, trains, temperature changes, injury, even surgery, other biologically active compounds, allergens, infections, and someone raised a question before about multiple infections. And that's actually a really good point because there's a possibility here that it's a cumulative effect, that it's not just a one hit, but maybe several hits over time will be enough to ramp up those aberrant receptors, if that's what's going on. And this is what the ion channel looks like, the 6 transmembrane elements, and between 5 and 6 here is the ion channel, and that's where the calcium flows through as the signal is amplified. And bear in mind that it is in every cell, in every tissue, in every system in the body to a greater or lesser extent, depending on how those receptors are expressed. And so when people say, well, CFS-ME is a neurological disease, well, we'd be inclined to say, well, yes and no. Yes, it's very important. I'll come to this in a minute of where they're located. But just about every organ system is likely to be affected in CFS-ME. So very important when you do research to have a hypothesis. It's a quaint custom which is based on actually proving that there is no relationship between the items that you're investigating. And it's a real trap to just accumulate data. And you should always say, what's your hypothesis? So if anyone presents data to you, the first question is, what's your hypothesis? And that's very critical, and that's what we've done. So, research question number 1. We identified those genetic differences, which I've just shown you. But what's the actual practical outcome of that? What actually happens to those ion channels on the cell? How are they expressed? So can we firstly identify these TRPM3 receptors on the cell surface in healthy controls to start with? And then what happens in ME/CFS patients when we look at the expression of those ion channels? And here we go, there's the bright cells and these are the dim cells. This is the expression of TRPM3, and this is a significant difference between healthy controls and CFS-ME. You can see the same trend here, but that's not statistically significant, but it's basically the same trend. And we would want to replicate that in more studies, but you get the drift of where this is going. So given that the job of the TRPM3 is to bring those calcium ions into the cell, the next step would be to say, what is going to be the effect? So do calcium levels in isolated NK cells differ between patients and healthy controls? And this is just some technical stuff. Here's the TRPM3 ion channel. And importantly, one of the major, or the major ligand for TRPM3 is pregnenolone sulfate. Pregas does activate other things in the body, but for TRPM3, that's the only game in town. So the ion channel effects here are regulated by Pregas. Now, the other thing is that to measure the calcium, you've got to be able to grab it at a point where you can measure the calcium in the cytosol, which is in here. But also calcium can be stored, not unlike a battery, in the endoplasmic reticulum, and that it's flowing in and out all the time. And this will lead us, and I won't spend too much time on it now, but if we have time, I'll come back to it at the end. It's a concept called store-operated calcium entry or store-operated calcium signaling. And what that means is that when the calcium comes into the cell, it can then regulate or transmit a message back to the expression of the ion channels on the surface to say, let more calcium in or don't let more calcium in. And that's a very, very important signaling mechanism. Now, if things were to go wrong with the ion channel here and it failed to bring in calcium, it's a bit like having your motor vehicle, the engine's turned off, a normal battery, but you leave the lights on overnight. And what that means is you're just going to deplete and run down the storage of power inside your battery. And it's a not dissimilar thing here, but we're talking about calcium ions instead. So what we can do is to block various changes that may affect that concentration of calcium in the cytosol by using these drugs called tapsigargan and 2-APB. And you're probably racing ahead of me now to say, well, if you can do that, does that mean that this is open for pharmacological intervention? And the answer is yes, it is. So there's a question here about, is the illness reversible? And let's just see whether we can follow that through. So this is just to show that when we use the different drugs like 2-APB and tapsigargan, that we can actually identify when we measure the calcium by the... when we measure the calcium that comes in here, we can see there's an identifiable difference between the patients and the controls. So there's the question. So are we able to influence the amount of calcium coming in through these damaged channels? We're not entirely certain just how they're damaged and what's going on there, but that would be really a great step forward. And when we started this whole process 10 years ago, we said to ourselves, what do we need to do? And we gave ourselves 3 aims. 1 was to discover the pathology, 2 was to develop a test, and thirdly, to identify suitable pharmacological agents. And we're pretty much at step 3. So this is how it works. So I've got 3 cartoons, so I'll just try and explain how this works. So here we go, there's the damaged, the SNP that codes for the gene of that particular ion channel, and they're the ones that I showed you earlier. And as you can see, there's a decreased expression of the TRPM3 receptor on the cell. So there's 2 questions. You know, how many receptors or ion channels are expressed? Has there been a reduction in that? And secondly, is there any change in function? So when we look at it, we, using certain techniques, we can say, yes, it's actually impaired expression of the ion channel to begin with. And then you'd say, well, what happens to the calcium? And the calcium is decreased, not only in the cytosol, but in the endoplasmic reticulum. And these biochemical pathways here are altered. So they lose some of their effectiveness and function by being impaired in that way. But the litmus test, as I said right at the beginning, is what is the effect on function? So you measure that in terms of lysis of the target cell. And at the moment, looking at that, you would say this is the situation in bright cells in CFS-ME patients. So we know that there is a decreased expression of the ion channel. We know there's decreased transmission of calcium, that is not good. Changes in the biochemistry here, and impaired lysis of the target cell. So then we do the intervention. As I mentioned before, pregnenolone sulfate is the primary ligand for TRPM3. So you would think, and this is the body now thinking, okay, I've got to react here and you know, bolster the function. So pregnenolone sulfate, when we add it, actually does increase the number of receptors, but nothing else changes. The calcium is still lowered in the cytosol and in the endoplasmic reticulum. And there's still the disturbance of the biochemical pathways. And the litmus test is it's made no difference, right? The rate of lysis of the target cell is still reduced. So in other words, if this was a normal situation in the body, then activating the cell with pregnenolone sulfate would actually reverse all of that and things would go well. But that is clearly not the case. So that's a very good indicator of what the pathology might be. So we do an additional intervention by using those drugs like thapsigargan and so on, where we can block loss of calcium out of the cell, and it's almost like artificially we'll prop it up a bit. So there's the pregnenolone sulfate, you've got increase in the receptors, and now the story has changed. You've got increased calcium expression, great, this is what we want. We then get a change in the expression of these pathways, and then the lysis of the target cell goes up. So this tells us that this situation is amenable to pharmacological intervention. So this is potentially a reversible illness. And this is how we measure the calcium. That's the calcium under the curve, and there's quite sophisticated gymnastics for doing that. So, summary, research question number 1. We can measure the function of NK cells in healthy controls and CFS-ME based on TRPM3 expression. Okay, so I don't want to dwell too much on this, but on the plane on the way over, I thought maybe if we looked at where the TRPM3 is expressed, it might help us to understand if that's going wrong. What has this got to do with the symptoms that patients experience? Now, the TRPM3 is primarily located here in the white matter of the central nervous system, so that's brain and spinal cord, where it has a critical role in store-operated calcium signalling. So this is in the white matter. Now, it's a slightly different, well, quite a different mechanism for calcium compared to excitable cells, which rely on voltage-gated channels. But these ones also influence whether the voltage-gated channels are turned on or not. So it's quite complex. But in their own right, they inhabit the white matter, and by doing that, they allow for a whole bunch of things to happen which are good for the health of the white matter cells. But importantly, they're also in the eye, in the iris, the retina, and the optic nerve, and the trigeminal and ciliary ganglia. Now, why that's important is often CFS-ME patients complain of pain in the eye, difficulty in seeing, and the TRPM3 regulates functions in the eye, in the iris, involved with accommodation, so your ability to see things. And by being expressed in the retina and the optic nerve, there's issues there about perception, of visual perception and so on. And the trigeminal nerve is conventionally regarded as a sensory nerve. It supplies the face. And the trick question you always get in an anatomy exam is, are there any motor functions of the trigeminal nerve? And the answer is yes, there's one. It's the stapedius, supplies the motor function to the stapedius muscle in the middle ear. So it has a very important role in the motor function of hearing. And the ciliary ganglia is important as a parasympathetic ganglia. It actually has several different nerve tracts, but being parasympathetic, its primary transmitter will be acetylcholine. So it would really be important to know, and you know acetylcholine is a very important neurotransmitter. It's important just about everywhere in the body. But also they're located in pancreas, so they have a role in co-regulation of insulin. And often CFSME patients really do complain of that metabolic problem, the crash, and eating sweet foods may be just one way to address it, but it may not be completely helpful. And the gastrointestinal tract, come to that in a minute, that's the thing. Cardiovascular system, and this is the story here, that the TRPM3 is actually co-located with one of the muscarinic, or maybe more, but we've done it in one, muscarinic acetylcholine receptors, and they're probably located on lipid rafts, where they are functionally and anatomically connected. So they really have a very tightly integrated set of functions. So there's a few other things here too, is that the two major intracellular transmitters in cells are calcium and cyclic AMP. Now, cyclic AMP is derived from ATP through an enzyme called adenylate cyclase. There's actually 9 types of adenylate cyclase, and numbers 1 to 8 are dependent on calcium. So you can see that there's a very, very tight link between not only calcium signalling but cyclic AMP signalling in the body. And therefore, together, plus the co-location with some of the acetylcholine receptors, you've got a very, very complex situation going on here. So I mentioned before this issue of accessing tissue, and up until now we've just done research into blood because it's accessible and patients help us with that. But we also wanted to know, well, what's happening in the gut? And there's a tissue that you can access through a gastrointestinal biopsy. So this is research question number 2. This is what we found. Don't worry about the top 2 there. These are simply negative controls. This is the healthy control here, CFSME here. So what you can see in the bottom left, TRPM3 in the healthy control, very robust expression. But when you look in the CFSME patients, you can see it's much more sparse. Not only is the expression considerably reduced, but with the eye of faith, you can see a little bit of disruption of the cellular architecture as well. So this is an incredibly important finding. And this was published yesterday, and this was Sonia's paper, and that was published yesterday. So, in summary, research question 2, TRPM3 expression is reduced in the gastrointestinal tract of CFSME versus healthy controls. So, research question number 3, what's happening with the muscarinic receptors? Now, firstly, can we identify them and characterise them and then interrogate them? And then can we identify those receptors in CFSME patients? But we also want to know, well, what's going on in the inflammatory situation in the gut, because there's been a lot of concern about the gut and possible inflammation as contributing to symptoms. And this is the story, that in healthy controls, a very robust expression of the muscarinic acetylcholine receptor called M3, and in CFSME, you would, I think, appreciate a decreased expression, and again, the cellular architecture seems to be somewhat impaired. So what this means is not only is TRPM3 impaired throughout the body, if we can say, well, we found it in NK cells and we found it in the gut, but it's also got an effect on acetylcholine receptors. And that's a pretty serious thing, because mostly people don't like interference with their acetylcholine receptors. It's usually doesn't turn out well. So the next question we then said, well, what's happening to the inflammatory status? Are we right by going down this path, or really is this a story about cytokines and inflammation or whatever? So
Done.
Done. interleukin-1 alpha is regarded as fairly important in triggering inflammatory pathways. And as you'll see, fairly robust expression here, but actually probably marginally reduced, the same or marginally reduced in CFSME. So that would tell us here that interleukin-1 alpha is actually reduced, the same or reduced, and therefore suggests it is not the primary pathology in CFSME. So let's see if we can get all the ducks lined up in a row. TRPM3 expression on the NK cell surface is reduced, calcium levels in NK cells is reduced, calcium entry into the cells, following application of the natural ligand, PregS, is reduced, but you can increase it with pharmacological intervention. And there's decreased TRPM3 in gastrointestinal biopsies, showing us it's probably decreased all around the body. That's what that's telling us. But importantly, there's also a reduction in muscarinic M3 receptor as well. So our conclusion at this stage is that impaired expression and function of TRPM3 ion channels affecting calcium signalling and calcium stores suggests the primary pathology of CFSME. At this point, I'd like to, there's Sonia, there's the wonderful team. I'd also like to acknowledge the chair of our steering committee, Mr. Ross Humphreys, who's here today, and Ms. Susie Brooks, who's also here, who represents the Alison Hunter Memorial Foundation, a very important charity that supported ME research for all this time. Thank you very much.
Don, please come and join us and we'll get the first questions to you here. Okay. Any questions for Don to begin with?
So does that mean then with the immunohistochemistry test that you would get now, that you will have a biomarker?
Have you been talking to him?
No, I don't even know.
Look, that's a really good question. And you've always got to be cautious when you answer questions like this because it's the amount of time involved. But if you go back and look at those changes, those SNPs, those TRPM3 changes I showed you, that would be the beginning of a biomarker. But the caveat that I really must put in here is that there may well be differences in the expression of those TRPs in different populations. You know, this is a population mostly Caucasian in the eastern seaboard of Australia, but it might be quite different in America or the UK or somewhere else. So we're just at the beginning of this story. So can I just answer your question and say there's a good probability we will develop a biomarker, and we're working on that with a pathology company at the moment. But can I caveat by saying, look, it's just the very, very first steps, but at this stage it's looking fairly optimistic.
Thanks, Don. Yes, question there, yeah. Rose, yes.
Yeah, it's Rose Bannings from New Zealand. I would like to ask, what's your next phase and what's the future going to hold? Because this seems to me to be a really major breakthrough.
Thank you. Okay, well, I think from the studies that we've showed there is that these damaged cells are amenable to pharmacological intervention. And I think that's the key thing to remember. So by now embarking on a very huge task of isolating various drugs and drug derivatives that have that same beneficial effect that we saw in the laboratory, but we also need to do that in the laboratory. We're not going to race in and start doing clinical trials until we have all the evidence we want from NK cells in the laboratory. But, and that'll be an incredibly tedious and difficult process because there's probably tens of thousands of drugs and drug derivatives that we're going to need to look at. But that's our next step. So,
okay, any other questions for any of the other speakers or Don again?
So you demonstrated some pharmacological intervention that altered the function of a cell successfully. Do you anticipate there being differences between cells from a single person, so that although there might be a sort of combination of chemicals that works in some cells, it might not work in others, and how do you sort of anticipate overcoming that challenge?
Yes, yeah, good question. And that's absolutely correct, because changes in our genes are so individual. You know, we all have them, we're all different, and that you know, may have been 20 or 30 patients. I just can't remember how many times we've done that one, but the point is that if you just take one patient, their SNP profile may be just a little bit different from somebody else's, and you can't say that one drug fits all. So what's really tricky here is the concept of personalised medicine, where you've really got to diagnose each case as it comes based on the profile of its SNPs in those ion channels, and there may be subtle differences between drugs and drug derivatives that actually means you're going to have to develop a kind of like a cocktail approach to each patient. And that's going to be huge. I mean, the logistics and the science of that is diabolical, but that's probably the direction we'll go. Thanks, George.
Well, thank you very much, all three speakers, for your contributions, and I hope you've enjoyed it.